Hi, everybody, and welcome back to part 2 of this week's lecture. What we're going to be looking at today is how to do Punnett squares and how we can utilize Punnett squares in order to predict the percentages and outcomes of the following generation. So let's go ahead and get started. What we're going to be looking at is the monohybrid genetic crosses. We're going to be looking at two specific examples. One of them where we're going to be taking two purebreds cross with each other, and then the second we're going to be looking at two hybrids cross together. Now, before we start, there's a couple important things we need to kind of recap and remember. One of the very first things is exactly what a phenotype and a genotype is. So remember, pheno, physical, what we can physically see, and geno, genetics, what the letters are going to be. Now, that's going to play a very important part when it comes to doing Punnett squares, because we're going to have to utilize and do something known as a phenotype ratio along with a genotype ratio. So with that being said, let's go ahead and get started. So you can see over here, if we go ahead and take a purebred cross with a purebred as well, you can see that we're taking big P, which is our homozygous dominant. Remember, anytime we have a capital letter, that's going to be our dominant gene. And we're going to go ahead and use that same letter, but a lowercase to represent a recessive gene. So in this case, homozygous, homo meaning the same, dominant meaning two big letters. So in this case, homozygous dominant meaning two big letters. You can see that over here, genotype is big P. We have homozygous recessive here. So homozygous, remember, meaning the same, recessive meaning little. So in this case, homozygous, same, recessive, little. In this case, two little letters. So we have our phenotype physical, which is going to be the color purple. And that's going to be big P. And then our phenotype for our second one is going to be white. And then our genotype is going to be little p, again, meaning recessive. So you can see over here that in our parental cross, we're taking big P cross with little p, and then we're going to end up with this F1 generation. Remember, our F1 generation is going to be the first generation from our parental. So you can see over here that we're going to have something known as a hybrid, which means that it carries both of the genes. In this case, we have big P, one half from this parent, and we have little p, one half from this parent. And this is going to give us a heterozygous. Hetero meaning different. So in this case, we have different letters. In this case, we only have two, big P, meaning that we have big P, little p giving us heterozygous. But our phenotype is still going to be purple. Although we do carry that recessive gene of white, we still have a dominant gene. In this case, it's going to be purple. So in this case, remember, dominant is always going to show over the recessive gene. So in this case, our phenotype is going to be purple. So let's see exactly how we can do this and how we can calculate and see the percentages and the chances of these certain offspring. So you can see over here that we're going to utilize something known as a Punnett square, which is this right here. Now, what we're going to do, one of the very first things we should do is we're going to be taking our P1, parent 1, and then our P2, parent 2, and placing them over here. Now, what we have to do is we need to segregate these or split them in half. So again, our cross is going to be big P, cross with little p. We have our P1 and then our P2. Over here, you can see that we're placing, in this case, our P1 or our big P up on top, and then our little p off to the side. Remember, we're segregating these. And these are going here and here, and these are going here and here. So now that we have this Punnett square, all we have to do now is we need to go ahead and populate or fill these boxes. So the way that this is going to work is we're going to go ahead and take this P and we're going to drop it down into this box here. So we have one big P. And then we're going to continue to drop this down into the second box, which is going to give us a big P here. We're going to do the same thing with this. Bring this down. So we have a big P. And then bring it down one more time, big P. So now you can see that we've dropped our P1, big P, into each one of our boxes. Then we're going to do the same thing. Let me go ahead and do that with a different color. Let's do blue right here. And then we're going to go ahead and do the same thing. We're going to take this and bring it into this box here. So we have a little p, bring this into this box here, little p, same thing with this, little p, and then over here, little p. So again, you can see that we've taken our P1 and we've brought both of these down, and then we've taken our P2 and we've brought in both of these over as well. And now we can go ahead and do our analysis and identify our genotype and phenotype ratios. So let's go ahead and do our genotype ratio. So I'm going to put a G for geno right here. And all we're doing here is we're going to take a look at our Punnett squares, and we're going to see or identify all of the different types of genotypes or the different combination of letters we can have. So in this case, we'll start off with this one here. Let me go ahead and change that color one more time to make it easier to see. So you can see we have one combination of letters here. So we have a big P, little p. You can see this one is the same. This one's the same as well, and this one's the same. So in this case, for our genotype ratio, we have big P, little p, and we have 4 of them. So in this case, our genotype ratio would just be a 4, or we can break that down into a 1. And what this is representing is there are 4 opportunities for you to have big P, little p, or a heterozygous cross, or a 1, meaning that there's only one selection out of all of these, which is going to be big P, little p. Another way of saying this is we have a 100% chance of heterozygous big P, little p. The next thing we need to do is we need to go ahead and look for our phenotype ratio. So remember, physical, what we can physically see from this. So although we have a dominant gene here and a recessive gene here, physically, what we're going to be seeing is the color purple. And the reason why is because we have a dominant gene over here. So it doesn't necessarily matter what this gene is, this dominant gene is going to be showing. So in this case, the 2 different colors that we could have would be white and purple. And then we count how many of these are going to be purple. So we have 1, 2, 3, and 4. So we have 4 of them that are going to be purple, and we have 0 that are going to be white, meaning that we have a 4 chance of it being purple. We can reduce that down to 1, or we can say we have a 100% chance of all the offspring exhibiting the color purple. Okay, and this is what that's going to look like, a little bit more cleaned up. And then here are the genotype and phenotype ratios right here. Again, you can see genotype ratio, every single one is going to be heterozygous. You can see we have a dominant gene here and a recessive gene over here. And then the phenotype, physical, what we're physically going to see. In this case, the physical trait is going to be color. Every single one of these is going to be purple. So this is our first example of taking a purebred purple and purebred white. A couple things I just want to touch on before we move on to our next example. Remember, purebred means homozygous. It's just another way of saying that because, again, it means that they're the same. And then hybrid means different, or in this case, hetero. And then when it comes to homozygous, we have 2 different types. Homozygous dominant. Remember, homozygous, the same, dominant meaning big. In this case, we have 2 big letters. Same thing, homozygous, the same, recessive, 2 little letters. And then heterozygous right here, meaning different. In this case, you can see we have 2 different types. All right, so let's go ahead and take a look at our second example. In this case, we're going to be taking a heterozygous crossed with a heterozygous. So what we're going to be looking here is we're going to go ahead and take our P1. In this case, we have heterozygous crossed with heterozygous. So we're going to go ahead and place these over here. So I'm going to go ahead and do the first one in blue. So big P, little p. And then remember, we're going to drop these down. So this one's going to go down this way. Big P. This one down this way. Big P. And this is a recessive trait. So whenever we're writing these, we always want to put dominant first and then recessive second. So in this case, we're going to put a little p here. and then bring this down, and then we have a little p here. Then we're going to go ahead and cross our second one right here, our P2. So we have a big P here, and then we have a little p here. We're going to go ahead and cross it over to this box. So we have big P here, cross it over to this box, big P here. And then remember, this is little p, cross it into here, little p, cross it into here, little p. And then now we're going to go ahead and do our genotype ratio. So I'll do that in green. So we have our genotype ratio. So remember, in our genotypes, we're just looking for all of the different types of letter combinations we can have. So we could have a big P, we can have a big P, and then we can have a little p. So all we're going to do is we're going to count how many big P's we have. So looking at this, we can see we have one big P. So that's it. So we have one big P. We're going to look for big P, or in this case, heterozygous. You can see we have one here and one here. So I'm going to go ahead and circle those. So we have two for those. And then finally, how many little p's do we have? Looking at this, we have one over here. So in this case, we have a 1 to 2 to 1 ratio when it comes to our genotype ratio. Again, we have one chance of it being homozygous dominant. So I'm going to put HD and 25% chance of it being homozygous dominant. We have two, or in this case, 50% of it being heterozygous. So I'll put HE, and we have a 50% chance. And then looking at this, we have a 1 chance, or in this case, 25, of it being homozygous recessive, 25% chance of that happening. So again, this is going to be our genotype ratio when it comes to our Punnett square. Next, we're going to go ahead and take a look at our phenotype ratio. So remember, pheno is going to be physical. So in this case, we have two options of color. In this case, we have it either going to be purple or white. So in this case, we're going to go ahead and take a look at this. So remember, when it comes to our phenotype, as long as there's one dominant letter, that is the color that's going to show. So in this case, if we have a big P, and then it could be either big P or little p, it's going to give us purple. And then the only way for us to get white has to be two little p's. So we can go ahead and take a look at either our Punnett square or our genotype ratio to kind of help us identify this. I'll go ahead and show you both ways to do so. So again, for purple, we're looking for a big P. So anytime we see a big P, it's going to automatically be a purple flower. So in this case, we can see we have 1, 2, 3 that exhibit those dominant features. So in this case, we have 3 that are going to be purple. And then you can see we have 1 right here that has both recessive. So we have 1. In this case, we have a 3 to 1, or in this case, a 75% chance of it being purple, and then a 25% chance of it being white. The other way we can look at it is our genotype ratio. You can see we've already kind of done that here. So we can see we have a big P here and a big P here. So we'd add these together. 2 plus 1 gives us 3. So 3 of those would be purple. And you can see we have 1 little p, meaning we have one of them that would be white. Okay, so this is kind of how you would do this type of cross whenever you're taking a heterozygous crossed with a heterozygous. Okay, and then let me erase all of this and show you the typed version that's a little bit cleaner. There you go. All right, so that kind of concludes how to do Punnett squares. Let me actually go back real quick and just kind of recap how to do a Punnett square with this over here. So one of the very first things you want to do is identify your parent generation, or in this case, your cross generation. So in this case, we're taking F1 crossed with F1. So we know we're taking this organism. We just need to figure out what our phenotype, our genotype ratio is. I'm sorry, genotype. So our genotype is big P, little p. So we have one dominant, one recessive, cross with big P, little p. Go ahead and place those. We can place these either places. So big P, little p, big P, little p, and then we'd go ahead and cross these down, bring these down, bring this over, and then we would go ahead and take a look at our Punnett square in order to do our genotype ratio. Remember, geno is the letters. So how many different letters do we have? In this case, or letter combinations, in this case, it would be big P, big P, little p, and then finally little p. And then our phenotype ratio, physical, what we can physically see. In this case, we have two different colors. We have purple, and then we have white. And then we would just go ahead and take a look at this Punnett square in order to answer our genotype ratio along with our phenotype ratio. Okay, so this is the part of the lecture where you're going to go ahead and test your knowledge. So as always, I'm going to go ahead and give everybody about 5 seconds to pause this video and give these a try, and then we'll go ahead and check these out. All right, welcome back. Let's go ahead and see how you did. So number 1, what genetic term could be used to describe the physical appearance of a purple flower on a plant? Remember, physical, pheno, P. It is the phenotype. Describe this genotype. We have a big A and a little a. So we have a dominant and recessive. So remember, we have two different types. Because they're two different, we have heterozygous. If they were both the same, then that would be either homozygous dominant if they were both capital, or homozygous recessive if they were both little. In this case, because they were different, it's heterozygous. Describe this genotype. We have two big letters, big B, meaning we have two dominant and they're both the same, meaning we have homozygous dominant. And in this one, it's the opposite. We have two little d's. So both of them are exactly the same. So we know it can't be heterozygous. We just need to look at the size now. They're too little, so we know they're not dominant, meaning that they are homozygous recessive. If an individual has a dominant and a recessive allele for gene X, that individual can be described as a heterozygous. If they have a dominant and recessive, so if we were to say dominant was the letter A, it would have a big A and then a recessive a little a. Again, two different, dominant, recessive, giving us heterozygous. A monohybrid cross is defined as a genetic cross that occurs as a result of two true breeding parents that differ in one trait only. And then the Mendelian genetics describes here are concerned primarily with the genes that are controlled how many traits. In this case, one. Remember, we're only dealing with a monohybrid, so just one. All right, so we're going to go ahead and take a look at this Punnett square. So in this case, we're going to be looking at widow's peak versus straight hair line. So just like before, I'll give you about 5 seconds to pause this and give this a try. All right, welcome back, and let's go ahead and check this out. So it says hairline, widow's peak versus straight hair line. So we're going to be performing a genetic cross between an individual who is homozygous dominant. So right there, we know homozygous. They're both going to be the same, dominant, two big letters. So we already have an idea of what our first is going to be for widow's peak. So we know our parental phenotype right here, our P1 is going to be widow's peak. And we know that this person, again, is homozygous dominant. So I'll just put HD just as a little reminder. An individual who is homozygous recessive for widow's peak. So right here. Oh, I'm sorry. Recessive for widow's peak, so straight hairline. So this person is going to have straight. Right here. And we know that they're going to be homozygous recessive. So the dominant allele is going to be widow's peak. We know that's going to be big H. And we know that the recessive for straight hairline is going to be little h. So what we're going to do is we know this person has a widow's peak. So right off the bat, we know they automatically must have a dominant trait right here. So we know they have a big H. So we just need to figure out what the second one is. And we know that this person has straight hair. Straight hair is going to be recessive, so we know they have two little H's. And reading back to this, we can see that the first person was homozygous dominant, meaning that they have two big H's. So now that we have our P1 and our P2, we're just going to go ahead and figure out what our Punnett square is going to be. So we're going to do our P1, big H, P2, little h here. We're going to go ahead and cross this. So again, we're going to bring this down through both of them. It's a big letter, so it goes first, big H, big H. We're going to do the same thing here, big H, and then we're going to cross these over. So little h, cross this over, little h, and just like that, we went ahead and completed our Punnett square. So now we need to go ahead and figure out what our geno and phenotypes are. I typically like to do the genotypes first and then do the phenotypes. So I'm going to do the genotype. I'm going to split this. So I'll do the phenotype here and then genotype here. So for a genotype, remember all of the different types of letter combinations we can have. So we can have big H, big H, little h, and then little h. So we're going to go ahead and see how many of each we have. So big H, looking through all of this, we have big little. So we don't have any of these, so we have a 0. Looking at this, every single one of these is going to be big H, little h, or in this case, it's going to be heterozygous. And then little h, we don't have any of them. So that means 0 for 0. Or in this case, we can just say that we have a 4 or 1, meaning there's only one choice when it comes to our genotype. And this also means that it is going to be a 100% heterozygous. When it comes to our phenotype now, since we did our genotype first, we can get a pretty good idea of what our phenotype is going to be. So looking at this, we don't have any of them that are going to be homozygous dominant. So that means none of them are going to be homozygous dominant. We don't have any that are going to be homozygous recessive. So in this case, meaning we don't have any straight hair line. So in this case, for our phenotype, we have a 100% widow's peak. So we have 4 that are going to be widow's peak, 0 straight. So in this case, we also have a 100% chance of it being widows. Okay, and then I'm going to go ahead and erase everything on this slide to show you that as well. All right. Here's another one. We're looking at orange versus white hair. So I'll give everybody 5 seconds to pause this, give this a try, and then we'll check it out together. All right, welcome back. Let's go ahead and do this one. So it says hair color trait. That's what we're looking at. We're looking at orange versus white hair. So we're going to perform a genetic cross between an individual who is heterozygous. So remember, hetero means different for orange hair. Right there, we know that that's going to be our phenotype. So we know this is our geno, we know this is our pheno, and an individual who is homozygous dominant for orange hair. The dominant allele is going to be O. So we know O is going to be dominant for orange. And then we have our recessive allele, little o, which is going to be for white. So little o, or 2 little o's for white. So we're going to go ahead and draw that Punnett square and see what the geno and phenotype ratios are going to be. So let's go ahead and check this out. We're going to do our cross. We have one that's going to be heterozygous for orange. So we know one parent's orange. And in this case, hetero. So I'll just put HE for hetero for that, for me to remember. And then we're going to look for the second person for orange hair. So we know that this person's orange as well. And in this case, they're going to be homozygous dominant, HD. So now we're going to go ahead and take a look at our parental genotypes. So in this case, our first person's heterozygous for orange hair. So hetero, remember, means different. So we have one big O, one little o. And then for our second parent, we have homozygous dominant. So remember, homozygous means the same, dominant means the same. So in this case, big O, big O. So now that we have this, we're going to go ahead and put our cross together. So we have one big O, one little o, one big O, and one big O. And then we're going to go ahead and do our Punnett square. So remember, this one is going to get distributed down to these two. So big O, big O. This one is going to get distributed down here. It's a little o. Remember, whenever we're doing our Punnett squares, whenever we're writing this, big letter goes first, little letter goes second. So in this case, we have little o here, little o here. Then we're going to take this and cross this over here. Big O, cross this over here. Big O, big O. Now that we've completed our Punnett square, we're going to go ahead and do our genophenotype ratios. So we have our pheno here, and then we have our geno here. Again, I like to do the genotype first, but you can kind of do this however you like. So we're going to look at all the different letter combinations. So in this case, we could have a big O, big O, little o, and then two little O's. So now we're going to go ahead and check this out. So we're going to go through our first one. How many big O's do we have? Right here, we can see we have one big O, two big O's. So now we have two big O's. And then we're going to move on down here. How many big O, little O's do we have? Looking at this, we see we have a big O, little O here, big and little O over here as well. So we have two over here. How many little O's do we have? We don't have any. So that means we have a 0. So in this case, we have a 2-to-2 ratio. This can be converted down to a 1-to-1, or this would be about a 50% chance of it having either homozygous dominant or heterozygous. Now we're going to take a look at our phenotype. And we know that big O is going to give us orange. And then we need two little O's that's going to give us white hair. So going through this, since we're taking heterozygous parent crossing with a homozygous dominant, you can see, remember, anytime we see a big O, it's going to give us orange hair. So in this case, we have big O here, big O, and big O. So in this case, we have orange and we have white. In this case, we have 4 of them that are going to be orange, 0 that's going to be white. So in this case, we would have a 100% chance of it having orange hair and a 0% chance of it being white. All right, and that is going to conclude part 2 of our genetics and inheritance. So again, as always, if you do have any questions, comments, concerns, please feel free to reach out. Until then, I look forward to seeing everybody in part 3.